Hello and welcome to EGR362 Developmental Biology for Engineers. Today, we will be going over the following. Developmental bio, of course. Which is about half genetics and half developmental. Technically, genetics and developmental bio are different. However, they are intricately linked. So they're separate, yet they cross over quite a bit. So first, let's get started with genetics. So genetics is the study of hereditary and the variation of inherited characteristics. So parents pass down traits or appearances to their offspring, in addition to species reproduction. So let's write that down, obviously. Study of heredity, and the variation thereof of inherited characteristics. Which puts a lot of traits in fair game. So everything from hair color to height to eye color to even some personality traits, obviously disease, some metabolic traits or behaviors, all kinds of different aspects go into genetics. And genetics implies study of genetic material. Now, when I was in undergrad, I took a genetics class, and not just material. We as engineers have material science, but not the tiny nucleic acid types. Genetic material, thank you. So in my genetics class, it was one half like life sciences, very intricate detail of how the enzymes replicate the DNA, and then how the DNA would be passed on genetically or passed down from parent to offspring, either in plants, animals, reptiles, birds, etc. But the other half was like trait inheritance, which gets very complicated. And in addition, I took a human birth defects class, which really went really heavy into how humans inherit different traits. So I'm going to give you about that much of it. I'm going to cover pretty broad concepts here. There's going to be a little bit more memorization than other aspects of it, or aspects of science, just because the different terminology is really specific to human inheritance. But like embryonic biology, it's a whole class. It's a whole academic class, but it's also in a whole class of categorization. So let's move on. The base unit for all genetic material in the more functional sense is called a chromosome. And let me check my audio levels. Okay, we'll try this. So the fundamental base unit of the genetic classification would be the chromosome. And it's wrapped long-stranded DNA. This is also called chromatin. Now, chromatin, a chromosome, sister chromatids, are all referring to DNA. The details are different, but the genetic material itself is the same. Humans have 46 of them, 46 total. Which work out to be 23 pairs. Obviously, because math. If I were to break this down further, 22 of these 23 pairs are called autosomal. Or autosomes. This is referring to body chromosomes, which means that they encode for proteins related to just human or mammal traits. I have hair, I'm this tall, I have muscles, a nervous system, a GI tract, I can reproduce, I can see out of eyes, that sort of thing. Whereas the one other pair of chromosomes are the sex chromosomes. And more specifically, they're the sex determination chromosomes. And they are referred to as, you can either have an X or a Y. And because it's a pair, there's two of them. Two X's mean female, and XY means male. Now, there are a whole host of genetic disorders that either add an X or add a Y, or take out one of them or leave the other. It's very broad. Those are rare. I'm only going to cover what, quote, normal, unquote, happens. So, interestingly, females can only pass on X chromosomes. Whereas males can pass either. I don't know what you do with that, but that's what the case is. Now, if I were to look at all 46 of these chromosomes and count the number of base pairs and the pairs of genes, they would look like this. So the total amount of genes in a human person is 21,000. Now, that's very high, obviously, but not as high as others, and not as few as others. There are some kinds of rodents that have more, some have less. There's not really a pattern to it. It's just what it is. Now, if I were to count all those base pairs on all of these genes, total base pair count. And just to review, a base pair count would be CG or AT, would be roughly 3.1 billion base pairs. Now that's a lot. That's pretty wild. 3.1 billion. Interesting fact, the Human Genome Project I believe they're based in San Diego. The Human Genome Project made it their goal to completely sequence all 3.1 billion base pairs. And they did it, sort of. They sequenced about 92% of the genome. There we go. 92% of the genome was sequenced. Now, this region is called the euchromatic. Think of euchromatic as a synonym for interesting or non-repeating. The euchromatic region is what encodes for all 21,000 genes. Now the other 8%, we are the 8%. The other 8% is called heterochromatic. And they are found in centromeres and telomeres. And we'll go through what those are. Heterochromatic. Found in, of course, as I just said before, centromeres and telomeres. Now, in order for us to understand what those two things are, we have to actually draw a chromosome. So let's do that right now. And I will change colors. Now, if I were to draw a chromosome, they would look like an X, either capital or lowercase. Interesting factoid here. The Y chromosome is, when you look at it physically and compare it to the X chromosome looks like that, and then the Y chromosome looks like that. Very, very small amount of genetic material compared to the X. Do with that what you will. Yeah, small chromosome for the Y, big chromosome for the X. That's it. Anywho, so they can either be fairly large, like a uppercase X, or a lowercase x in size. So let's draw a chromosome. Part of it is just like this randomy X-looking material, or X shape. And that's because all of the chromosomes are wound in a semi-ordered function, or an ordered manner. The way that you are able to see what the chromosome shape is called a karyotype. Karyotype is it's like a physical mapping. Not really, it's like a physical snapshot of what the chromosomes actually look like. So, processing method or visualization. Of chromosomes. Now, this is really important in genetic disorder studies. For instance, if you have one extra or one few, or the X is one too many or one too small, like all of that really matters inside of the karyotype. So moving on to what a chromosome would actually look like. As I mentioned before, the centromere is a highly specific place. It's the center. Nothing gets past you at home, does it? This is called the centromere. And the centromere is what fibers bind to during mitosis. Mitosis being the cell type just the constant replication of the cell types, carbon copies. And that accounts for a piece of the 8% of the heterochromatin. Now the other part is a telomere, and that's the cap. There's little caps. And these little caps are repeating base pairs. So they're very long, but yet they're not so long. So these are the telomeres. And the telomeres are repeating base pairs that protect the actual the important genetic material during replication. I'm going to shorten BP for base pair. So they're repeating base pairs that protect the chromatids. Notice I said chromatids. A chromatid is one of the strands. So if I were to divide this down the middle, these both would be two chromatids, or sister chromatids. A chromatid is just one side of the X. And there was a knock at the door. Have no fear, no one's in here. So the telomeres protect the ends of the chromatids. Now, these are an interesting little phenomena. Telomeres are linked to aging. As mitosis occurs, some of the telomere is lost. And you might think, okay, whatever, I lost 5 base pairs. But over 80 years, you're going to lose a lot of the telomere. And telomere length is a function of age. So as age increases, telomere length decreases. Except in some very specific cases. So as the telomeres shrink, that's usually an indication of age, but it's not necessarily linked to your actual age. I, as a 26-year-old male, could be looking like my cells could actually look like an 80-year-old under the right conditions, which is a little weird to think about. Like, you yourself are not old, but you, not conscious you, is really old. You philosophers in the room should think about that and then write a book, as philosophers do. Moving back to telomeres. So there is an enzyme that can repair these, and the enzyme is called telomerase. Similar to DNA polymerase, the telomerase repairs all of the telomeres. It can actually add back in those base pairs that were lost. Telomerase can repair the telomeres. And in practice, that would look something like, so they just have like 1 million A's. And then the telomerase would just add in more A's. So then it would wrap around the whole strand. Now, as the great philosopher Ned Flanders says, the next part is a real noggin scratcher. Because telomerase is promoted in 2 very different things. The first one being exercise and meditation, in addition to cancer cells. So telomerase builds the base pair lengths, and that's a good thing if it's coming from exercise, because that means your metabolic health will be younger, you'll be more calm, you'll be stronger, all a great thing. However, in cancer cells, it's a great biomarker to detect cancer. It's called a biomarker for cancer detection. Because telomerase makes the cells immortal. Immortalizes the cells. Now, that's not a good thing. But then again, it is a good thing to have it in your system. So as I referred to before, this is kind of a noggin scratcher. Because if you have too much, then you have cancer. But if you have none, then you get old, which then would possibly lead to cancer. So then it's just a very strange loop. We don't know how to fix this. We don't know what the right amount is. We don't know if any of those, if that's a good thing. If telomerase is a spy for like the cancer mafia, very interesting. So more research has to be done there. Okay. Moving on to further classification of genetic material. Now, if I were to look on a karyotype, some of the genes would be dark. Or not some of the genes. Some of the regions on the chromosome would be dark. And part of that has to do with genes, like what base pairs are where. Real quick. Sizing. The chromosomes can be 0.2 micrometers all the way up to 20 micrometers in height. Interesting. So a gene is a character on Bob's Burgers who says great lines like, "Queen Latifah, give me strength." But that is not what, there's a better definition of gene, or rather a gene. It's a specific region on a chromosome that encodes for a protein monomer. On a chromosome. And the word monomer means just single unit, like one, just one of something. That encode, and many of these monomers will make a polymer, which can be a repeating chain or different chains. The word polymer means repeating chain. But you can also think of proteins as being monomers. Okay. Monomer. And when I use the word monomer like before, I mean protein unit. And the protein units will carry with them inherited processes, such as DNA polymerase, its job, replicating DNA, so on and et cetera and so forth. Now, related to a gene is something called an allele. An allele is an alternate form of a gene. And these are commonly denoted as an uppercase letter and a lowercase letter. These were made famous by the fellow who developed the Punnett square. Mendel, I believe. Yeah, I believe it was Mendel. So if I were to cross, well, this was his work. He worked with peas and lilies, flowers. So if H encodes, I'm going to use B. If H encodes for round, and I don't know exactly what I started with, I can just cross-pollinate 2 peas and see what I get. And if they all come out, and rather if 3/4 of them come out round, and he did quite a few samples, so they actually came out to be about 3-to-1. If I were to cross-pollinate all of them, the genetic square, it's called the Punnett square, the genetic square would look like this, where the green are the round peas, and the blue circle, triangle, shouldn't use round, that would be confusing. So it's called the recessive trait, meaning lowercase, lowercase. That's called the recessive trait, which recessive only is broadly used for traits or genes that are not encoded or that don't show up in a phenotype, versus the green, which is the dominant. Now, if you were paying attention, I used a new word here, and that's phenotype. Phenotype is the physical expression of a gene. Excuse me. So the dominant gene and the recessive gene both have separate phenotypes. In this example, the dominant was a round pea, and the recessive was lumpy. And that was Mendel's work with peas. Very famous, very good scientific literature there. But he lived back when he was a monk and he was a farmer, interestingly enough. So dominant just means that another way to think about dominance is that the phenotype will override if a dominant gene is present. That's what the phenotype will be. That's what the phenotype will show. So so we can do an if statement. If present, if big H is present, then round. Else, lumpy, and end. Ta-da! Genetics turned into if-then-else statements. If that's not engineering physiology, I don't know what is. All right. Now, we don't use Punnett squares to describe human heredity. Instead, we use something called a pedigree. And a pedigree is a chart with shapes that describes whether a trait or disease or condition is present. And they look like this. I'll change colors. Whoops. So a pedigree would look something like this. Males are square, and females are round. And they have offspring. So you just draw a line and count how many kids there are. If I were to do my family, this would be me, my sister, and my brother, my younger brother. Now, notice I gave myself a little bit of space here. That's because my sister is married to my brother-in-law. And then rinse, repeat. They don't. None of us have children, so this would be the pedigree for my family. Ta-da! That's it. Now, let's pretend that some. Well, okay. Here's something. Let's pretend I want to see who has blonde hair in my family. So if I use red for, quote, the affected individual, then I would just shade that in. So I obviously have blonde hair, so I would shade in myself. In addition, my sister has blonde hair. Furthermore, we have the same hair. She's a little bit shorter than me, but we have the exact same doppelganger type hair. My mom and my dad, interestingly, do not. They have more brown hair. My mom kind of has blonde hair, sort of. My grandmother, on the other hand, very vibrant blonde hair at one point. So, if I were to create a little guide, it would look like this. So shaded in areas are affected individuals. Can you guys read that? If you look really hard, you can read that, but I'll try to write bigger next time. So an affected individual will be shaded. And then a couple more interesting facts. If I were to write twins, then they would look like this. So single branch from the parents. And then depending on if they were identical or not, or if they were both male or both female or one or the other, it would look like this. So one branching off to the other. If they are what is known as monozygotic, then they would have a line right here. Monozygotic, which is just a fancy word for identical. Now, if they were dizygotic, they would look like this. Just one branch with no line, empty line. Dizygotic, which is another fancy word for separate. Okay. All right, let's move on. I'm just going to erase this part of the board because I'm still going to reference the pedigree. So one way humans inherit traits are what are called sex-linked, which is a fancy word for like all males or all females inside of a family. So sex-linked traits, not lined traits, that would be silly. Linked traits is a fancy word for like all sons or all daughters, something like that. So let's pretend that this is a made-up family and there are no affected individuals to start. But then let's retroactively go back in time and say that only the women in my family are allowed to have blonde hair. So that means my sister and my mother, or it would depend on her parents, which unknown at this point, which is what happens a lot of the time with pedigrees. It's like, what do they look like? I don't know. They still wrote with feathers back then. Who knows? So you just have to work with what you're given. So all sons or all daughters would look something like that in the pedigree. Interestingly enough, there are mutations which can propagate through generations. Going back to dominant versus recessive. Dominant traits in general are present in, if one were to propagate, they would be present in all generations. In contrast, if it's a recessive trait, then the phenotype would tend to skip generations. And that's fairly intuitive. So you can really tell that easily in a pedigree. It'll be like all color, no color, some color in the grandchildren, something like that. Okay. Moving on. I'm not going to spend a whole lot of time on mutations. I've talked a little bit about them in the DNA replication lecture, but they're worth noting. They're usually changing a codon inside of the protein coding mechanism. Which means that base pairs are changing. Now, there are 3 real types of mutations as a net result. So the first one is deleterious. You see a lot of these kinds of mutations develop inside bacteria because their generations, or their generational count is like 10 to the 3rd on the low end. Like they can just replicate like crazy. More than bunnies, believe it or not. I know, crazy. So a deleterious mutation would be negatively impacted. Negative impact. Which in a bacterial sense means that they die. A gene isn't coded properly, and then the protein doesn't happen, and then that is one of maybe 15, 20, 100, 1,000 genes, and that's really important, so they die. Second one, and this is much more common for anything else. Or the second most common is neutral. Nothing happens. Nothing. And the third one, a positive benefit. Now, I am not an evolutionary biologist, but if I were to play an evolutionary biologist on the internet, a positive mutation would yield some kind of usable trait or usable protein. or usable metabolic reaction, etc. I'm still unsure about the real effects in human genetic mutation. Bacteria, it's pretty obvious. So the most obvious case is antibiotic resistance. Go look up a video that's like E. coli, real-time evolution, something like that. What the scientists, I think it was at Harvard, what the scientists did was make a big plate, and they loaded up one side full of E. coli. And the goal was to get the E. coli from one side of the plate to the other. However, they made the track laced with different kinds of chemicals. There was thumping, okay. Chemicals meaning antibiotics or medicine or very difficult barriers to get past. So what would happen is they would like have a bunch of them go, and then they'll like a few of them make it to the next one, but then only a smaller strand would make it over, and then so on and so forth until, so you get like massive decreasing. That looks super messy, but go watch the video, it's really cool. So you get such a tiny actual result. You get such a small amount with what you started with, compared with what you started with from the beginning of the E. coli culture numbers. Fascinating stuff. I don't know what it means for humans, but at least for bacteria, super interesting to watch. All right, we're going to really shift gears now and talk about human developmental biology. I took a class on this, and I had to memorize so much stuff. I learned how to do it, and the lab was super interesting. I had really good TAs, mostly because I hung out with the cool kids, and the cool kids knew the TAs, like outside of class. So I was in the cool kids club without actually trying, which is the best way to do it. However, the class was really tough. But I learned a lot, and hopefully you learn a little bit about how stuff actually happens. So the first part in any developmental biology state is fertilization. So fertilization just means that a sperm cell, not a cell, but rather an egg, come together and make a zygote. Now, a zygote is just a fertilized egg. If I were to look at them under a microscope, the egg would just be a ball. But the sperm cell would have a pointed head and a flagellum. Now, in reality, the sperm cell is so much more small than the egg, it's unbelievable. The egg would be like this, and then the sperm cell would be like that. No, not even, it would be like this. Ridiculously small, comparatively. I am grossly over-exaggerating the size of this sperm cell. In reality, it's like smaller than my first pinky knuckle. Cut that in half, and then cut it in half again, and that's about right. Very, very, very small. So once the zygote forms, you get something called a blastocyst. blastocyst, which sounds something out of like a 90s cartoon super marketing campaign. However, a cyst just means a clump of cells. But this clump of cells has a very interesting characteristic. It just looks like a ball. The zygote will go from one cell to, this is a terrible marker. The zygote itself will go from one cell because the sperm and the egg all have half the number of chromosomes. They each have 23. Together they make 46. And there's my good marker. So this zygote is one cell. And then we go through a bunch of multiplication numbers and tables, and then end up at a blastocyst. Blastocyst has many cells by number. But the important thing here is region. Anytime in developmental biology, region is really important. The region of a cell, not the region of a cell, the region of a blastocyst, or later what's called a germ layer. Location is everything in developmental biology. Now the best example of this is this little shaded in area right here. It has the best name ever. It's called the inner cell mass. Very creative. Hollywood I don't know. But the inner cell mass is exactly it's very, very interesting. Mostly because it secretes a hormone. And this hormone is called H_CG. And HCG stands for human. I always get this word wrong. Chorionic. Gonadotropin. HCG. And human gonadotropin is a hormone that is only present while the woman is pregnant. Only present during pregnancy. Now, that's really useful from an engineering standpoint. If I'm trying to find out a state of something, I'm going to look for a very specific molecule. For instance, if a piece of metal is hot, if it's a dark color like it is normally under room temperature, I can't tell what temperature it is. However, if enough heat is applied to it, then it turns a different color. There's a whole lot more energy because of its exuding light. Therefore, I can tell color, or I can tell temperature by color. Similarly, in biological terms, pregnancy, I can look for very specific molecules that are only present during a state. And this is the hormone that a pregnancy test will look like. Now, it's really easy to make a pregnancy test. Just say, look, I need a color-changing piece of paper based on if a hormone's present. If it's present, then positive. Therefore, plus. If not present, then nothing. Excuse me. Now, only women can produce this hormone. If a man takes a pregnancy test and it shows up positive, that's not for this lecture. Go look up that on your own. But this is the hormone that I want you to come away with. HCG, human chorionic gonadotropin. It's present in pregnancy test measurements. Engineering physiology for you. All right, moving on to more about blastocysts. Blastocysts are inner cell masses. I'm going to change colors. The inner cell mass is also a source, I say a source of embryonic stem cells. Now, there's a few other places where you can get them, but these are the most able to divide up into every kind of cell type in the body. In the early 2000s, the president at the time, George W. Bush, outlawed all research on embryonic stem cells. And it's controversial, go look it up, make your own opinions. But this was a part of it. No work on embryonic stem cells. Another piece of a blastocyst is that this is what is implanted. Can you still see? No, you can't. I'll go over here. A blastocyst is what is implanted inside when a woman or couple is going through in vitro fertilization. IVF stands for in vitro fertilization. In vitro fertilization. So they fertilize, grow it in a cell petri dish, and then put it back inside of the uterus. And then hopefully that turns into an embryo, or what is commonly known in like lay terms, an embryo. Technically, this is an embryo. And that's why it's a source of embryonic stem cells, because technically the blastocyst is an embryo because it's fertilized. But there's a lot of political debate about like, this is a baby, no, this isn't, what's this, figure that out, I'm not gonna comment. All right, continuing on. So the different parts of an embryo, we're gonna go through what's called the germ layer. Remember back to a few minutes ago when I was talking about how it's all about location, location, location when it comes to developmental biology. So in what's called a germ layer is just a layer of cells. So this is a part of the different embryonic layers. Germ layer, just cell layer. But since this is on the way beginning end of the developmental process, the cells themselves are able to differentiate into different kinds of cell types. And that's an important term here. Differentiate. Its formal definition is, let me look at that. The process, trust the process. The process of a cell changing from one cell type to another. Related to that, another term is potency. Potency is the ability to differentiate into new cells. So the cell's ability to differentiate. cell's ability to differentiate. So if I were to say if a cell is pluripotent, that means it has many avenues to choose which cell type it's going to eventually end up becoming. These inner cell mass stem cells are extremely pluripotent. They can be anything at all. But the ones that I'm going to have you learn are based on the germ layer, where they actually are in the embryo. And there are 3 pieces to this. One is the endoderm. I need a little more space. Mesoderm and ectoderm. Sorry, I'm actually going to change. I'm going to go from the outside in. So I'm going to change endoderm and ectoderm. I'm going from outside in. So the ectoderm, those cell types, those cells will eventually turn into the following tissue types. The first is nervous tissue. The nervous system. In addition, tooth enamel. And third, the epidermis. So the ectoderm is the outer layer of the embryo. This is really general for a reason, because at each stage of pregnancy, the layers are in different places. And one cell type will be, or one piece of the ectoderm will be like over here and then over there. Like the nervous system includes the cleft in your lip, the pieces of lining in your mouth, some things in your hindquarters. So all of this is really mixed up together. So I'm just giving you the most general cases. Generally speaking about germ layers, these are the kinds of tissue types that you're going to get. So in the mesoderm, this includes all the blood and blood derivatives like neutrophils, immunity, etc. Muscle, smooth, cardiac, etc. And 3 is connective tissue. And the endoderm are the innermost germ layer within the embryo. So the endoderm will eventually, I don't really want to say encode, but they will differentiate, is a better word, into the GI tract. The endoderm is commonly known as the tract differentiator. So the GI tract, the respiratory, and the last one is the endocrine system, or endocrine organs. There we go. Time to take a sip of water. And that's a lot of developmental biology in a nutshell. I want to touch on stem cell therapies for just a few minutes. So I want to pay attention to this one right here. Connective tissue stem cells. All of these are considered stem cells, or at least in the embryo, or a layperson would call these a stem cell. Technically speaking, not all stem cells are equal. Some are more equal than others. And let's talk a little bit about that. The most common therapeutic type of stem cell is called the mesenchymal stem cell. or if you want to say it quickly, mesenchymal. Either pronunciation is acceptable. The abbreviation is MSC, thank you. Mesenchymal. Except there's a Y and not an A. Mesenchymal stem cells. And they are found in some very specific places. These are all of your therapeutic agents. Therapeutic, or a common thought is that they are regenerative. Regenerative therapy, regenerative in connective tissue, regenerative in brain, like function, regenerative in personality. Maybe not personality. Well, there's research. There's a lot. Okay. You can look up the regenerative properties. But I want you to know their locations, because these are the major therapeutic-based cells. And as the theme for this whole lecture is location, all about location. So the first place you'll find MSCs, or mesenchymal stem cells, are the bone marrow. I asked you a question about this on your last exam, assuming future me decides to have that question on the exam, which is it's a good question. Chances are good. The second place are cord cells. Now, this is not a power cord, but rather an umbilical cord. Umbilical cord cells. Third, adipose tissue. Now, what's adipose? Fat. Fat. And two more. First one is molar. Now, you might think of like millimolar, or the burrowing, furry rodent animal, but rather it's in the teeth. The teeth in the back of your jaw, about here, those are your molars. And it's the root of the tooth, teeth roots. Because teeth is the plural, teeth roots. And then the last one is the amniotic fluid. Now you might ask, where did that come from? Good question. I can actually add it onto here. It's another piece of the embryo. So the embryo would also include the amniotic sac, and the placenta. So the placenta, the embryo, and the embryonic sac all come from the blastocyst. And then this is where we get some mesenchymal stem cells. Some of them require a little bit more attention than others. For instance, the cells marked in green require very little preparation or post-processing. The cord cells, for sure. The amniotic fluid, absolutely. And interestingly enough, the molar cells. They don't need as much post-processing as the other two types of tissue. The other two types require some laboratory techniques, which is why I put a square. It's open. It needs some more post-processing, which can be spinning in a centrifuge, GFs, not girlfriends, they're singles, well, so to speak. GF meaning growth factor, or other new ones yet to be discovered. I suspect that they will be easier by the time in like 10 years, maybe 5 years, probably less. But these two types of tissues definitely need post-processing. Bone marrow, not as much as fat. Fat is the most, it's the easiest to get, but it's the hardest to de-differentiate. Very interesting stuff. So in conclusion, thanks for listening, and as always, I hope you learned something.